Welcome to this last episode of the EVOLTREE seminar series on polygenic adaptation. It's the last episode that we have planned for now, as it was a pilot series, sorry, that we wanted to try out. So we will be sending an evaluation survey after the series to all participants to also collect your feedback on it. But you will hear something about it later on. For now, I would like to welcome Neda Barghi today with us, and we are really honored and pleased that she accepted also our invitation. But Antoine will tell us more about Neda and will also tell us more about EVOLTREE network. So over to you, Antoine.
Okay, thank you, Eva. Hello to everybody. Well, I will be, as last time, I will give a brief introduction of EVOLTREE and then introduce, of course, Neda, who is our speaker today. Now, EVOLTREE is a European network of labs with strong emphasis on genetics and evolution in forest ecosystems, mainly on trees and associated species. In total, we are about 30 labs from 19 different countries, and this means about a few hundred scientists and students being members of the EVOLTREE of the network. The network is 15 years old. Initially, it was supported by the EU, but now is self-supported by its members. What are our objectives? We try to coordinate and share research efforts and resources at the European continental scale. And what our activities will be, we build and maintain common infrastructures, for example, monitoring plots, and also electronic resources as databases, interoperable databases. We organize training sessions in the form of workshops and summer schools, and nowadays, of course, webinars. We provide financial support for small-scale research projects, opportunity calls, and we maintain also networking activities. So far for EVOLTREE. Now today we have the pleasure to welcome and introduce Dr. Neda Barghi, our speaker of the last session of this polygenic adaptation series of seminars. Dr. Neda Barghi is a group leader at the Institute of Population Genetics at the University of Veterinary Medicine in Vienna, so-called VetMed Uni. Neda received her PhD at the University of Philippines in Diliman, working on the evolution of toxin genes in conus species using comparative genomics and transcriptomics. She did her postdoc at VetMed Uni with Christian Schlotterer, working on the genomics of thermal adaptation in Drosophila. Dr. Barghi is an evolutionary biologist investigating the genomics of complex adaptive threats in Drosophila. She's looking at the genomic signatures of adaptive evolution in response to biotic or abiotic stresses in Drosophila, of course. Her approach is mainly experimental evolution, where she implements evolve and resequencing studies. In her approach, she uses a wide range of methods, going from tools, pulsec time series data, molecular and phenotypic arrays, and also computer simulation to investigate the genetic basis of complex threats like thermal tolerance and body size in Drosophila. Now, to make a link with our previous speaker, Sam Yeman, last week, she's also investigating the genetic architecture of threats, looking at genetic redundancy in different populations of Drosophila. And I guess this is what you will be talking today. So I give the floor and the mic and the screen to Neda.
Thank you very much. Thanks for the invitation. I'm really happy to be here. And I'm looking forward to the questions and discussions that we will have. And thanks, Antoine, for the kind introduction. And I think it's been a very long time that nobody has pronounced my last name correctly, but you did it. I think it's just because you're French, probably. So thank you. So I'll share my screen then. Okay, so most of the things that I will be talking today is what I did during my postdoc in Christian Schlotter's group. And I've just started my own group less than 3 months ago. So some of the aspects that I took from the postdoc, I will be using it also in my research group, and I will be talking about it a little bit. And so it's mostly about polygenic adaptation in experimental evolution context in Drosophila stimulants. And although many people might work on natural populations, but I believe that some of the themes of or signatures of polygenic adaptation that we observe in experimental evolution can also be seen in natural populations, and I will be talking about it today. So one of the long-standing questions in evolutionary biology has been, how does adaptation work? And of course, in the field of molecular population genetics, adaptation is assumed to occur through large frequency changes at many independent loci. So beneficial mutations increase in frequency until fixation, and the consecutive fixations of beneficial alleles increase the fitness of the population. And if you look at the this mode of adaptation leaves a very clear footprint, which is the reduced genetic variation around the beneficial mutation. This footprint has been used in many tests of adaptation to identify regions that has undergone selected sweep. And of course, there are different flavors of selected sweep. The sweep can be soft, where the adaptive allele is present on multiple haplotypes, or both hard and soft sweeps can be complete or incomplete. In the field of quantitative genetics, adaptation occurs via under the infinitesimal model, where a large number of contributing alleles experience subtle frequency changes after a shift in the trait optimum. And that would result in changes in the mean trait of the population. And of course, unlike the footprint of selected sweep, the footprint of polygenic adaptation is quite challenging to identify, to say the least. So molecular population genetics and quantitative genetics have developed opposite views of adaptive processes, invoking either selected sweep or small shifts. But polygenic adaptation, which describes adaptation of quantitative trait, subtle frequency changes of very many loci, is normally, when it's normally contrasted with selected sweep, can be something in between these two ranges, because recent theoretical and empirical work have shown that even under polygenic adaptation, you can have large frequency changes. And also both sweep and small shifts are the extreme ends of a spectrum. So adaptation can actually occur through this spectrum. So most of the traits that have been characterized at the molecular level, normally with simple genetic basis, these are the textbook examples of selected sweep. For example, pigmentation in mice, lactose tolerance in humans, resistant to viruses, insecticides, and malaria in Drosophila and in humans. But also many of the adaptive alleles, adaptive traits are polygenic. They have this polygenic basis. we see a lot of studies of artificial selection experiments or quantitative trait loci studies in Drosophila, where large genomic regions respond to selection. But we also see in human GWAS studies that many of the traits, for example, human height, blood lipid level, or metabolic rate, they have very many contributing loci. So polygenic adaptation, which is adaptation of a quantitative trait, after it goes through a shift in its trait optimum. So during this adaptation, during this shift, alleles which contribute to adaptation increase in frequency so that the population would reach this new optimum. And during this adaptation, different stages would occur. So polygenic adaptation normally has different phases. So in the first phase, that these phases are depicted with these gray dotted lines. So in the first phase, selection is directional, and this is where when the trait mean adapts rapidly to reach the new optimum. In phase 2, shown here, allele frequencies change, but under weaker selection, but also under genetic drift. And in phase 3, this is where drift actually disturbs the allele frequencies beyond a critical point where the fate of alleles is determined, whether there's going to be fixed or lost. And of course, this pattern is different in small populations, for example, what we have in experimental evolution setup, because the second phase is mainly affected by drift, and drift is stronger in small populations. So disturbance of allele frequencies is much more in the second phase, and because of that, this second phase is actually much smaller. But also you can imagine the heterogeneity across contributing loci, including the starting frequency of them, can also determine the fate of them. For example, if the starting frequency is lower, they would be lost, for example, earlier than those that have higher frequency. So this transition between the second and third phase has a rough border depending on the genetic drift. One of the important characteristics of polygenic adaptation is genetic redundancy. When we observe non-parallel signatures of adaptation across different replicates. So for example, here replicate populations can reach the same trait optimum using different sets of alleles. This replicate 1 uses blue and green allele, and another replicate uses 3 other different alleles, but at the end, all of them reach the same trait optimum. But of course, you can imagine that the distance to the trait optimum and also the heterogeneity of loci in the founder population are also very important characteristics of polygenic adaptation. In fact, adaptation to the new trait optimum can also be achieved by a sweep-like signature, meaning that, for example, in this example, the intensity of color shows the effect size of alleles. So a population can reach the trait optimum by fixation of a large effect allele, or that large effect allele in another replicate can be lost, and subtle frequency changes of alleles with smaller effect sizes can result in adaptation of the population. And at the end, both replicates reach the new, the same trait optimum, but one of them, for example, has a sweep-like signature. And I think that experimental evolution has provides some opportunities that allows studying polygenic adaptation. One of them is replication, as I mentioned. To be able to really see genetic redundancy, you should have multiple replicates, and experimental evolution gives you this, this opportunity really well. The other one is to see the dynamics of allele frequency changes. And this is what you can have by having time series allele frequency data and also time series phenotypic data to basically see where the new trait optimum has been reached. And all of this can be provided in the context of experimental evolution. And before I make people who work in natural systems angry, I should also mention that there are also many natural systems that are also suitable for studying polygenic adaptation. For example, a lot of latitudinal or altitudinal clines in plants, in Drosophila, in humans can be used for studying polygenic adaptation. For example, Tibetan and Andean populations that have adapted to high altitude can be examples of two different replicates, for example. Or, for example, populations of cichlids that have independently colonized lakes, or populations of gopi in rivers in Trinidad that are exposed to high or low predation. And also, I think the best example would be stickleback that are present in multiple river systems, and it would be a great opportunity for contrasting marine and freshwater, lake and stream, or benthic and limnetic habitats. So, but it should be noted that all these populations, although these replicate populations, although they are exposed to this similar selection pressure in similar environments, but these environments are not exactly identical. And so the possibility of local adaptation still exists. So this is something that should be taken into account while working with natural systems. So during my postdoc in Christian Schlotter's lab, I analyzed a data set that was lab natural selection to a new temperature regime. So in that, we had a improved design compared to some other previous experimental evolution studies. In that, we had 10 replicate populations. And these 10 replicate populations were all identical. They started from 202 isofemale lines. These lines were collected from Florida. So we started from 202 times 2 sexes times 2 chromosomes with a lot of founder chromosomes in the population. So the available genetic variation was pretty high. And then we worked with Drosophila stimulans and not melanogaster, because stimulans has higher recombination rate compared to Drosophila melanogaster, and also it does not have long segregating inversions. So, and these 10 replicates were maintained in an environment which we call a hot environment, meaning that for 12 hours a day, they're exposed to a high temperature of 28 degrees where light is on. And for the other 12 hours, they're exposed to a cool temperature, which is 18 degrees, and then light is off. And we maintain the populations at population size of 1,000. And we had time series high coverage pool sequencing data. So we have time series data from generation 0 to generation 60 with intervals of 10 generations. So the first question that we asked was, have these populations evolved to this new environment, and have they changed compared to the ancestral one? So we performed some phenotypic experiments. So mainly we checked fecundity, metabolic rate, and body fat. And then we noticed that the evolved populations, which are shown here in red, are more fecund. They lay more eggs compared to the ancestral population. They produce more, females produce more carbon dioxide, and then they have less body fat. So in this regard, the evolved populations have changed compared to the ancestral one. And then we looked at the genomic data, and with this, we initially compared the pool-seq of basically the SNPs in the founder population to the data set at generation 60. So we compared allele frequency changes between the founder population and after 60 generations of an adaptation. So we use CMH tests to basically look for significant allele frequency changes across multiple replicates. And in this Manhattan plot, I show chromosome positions in x-axis and negative log 10 p-value of summary statistic of CMH tests in y-axis. So we have Drosophila similis has 3 chromosomes, chromosome X, 2 and 3, and chromosome 2 and 3 have left and right arms. So you see that there are a lot of regions of the genome that seem to be responding to selection. We normally call them peaks or chimneys. But of course, because the population size is small, drift is also strong. So there are some allele frequency changes that are solely because of small population size, because of drift. So we had to account for that. And normally what we do is just we perform simulations using the starting frequency of SNPs to see how much of the allele frequency change that we observe can be explained by drift only, and how much of it is more than what we expect only under drift. And this black dotted line that you see is a threshold of significance, meaning that all the SNPs that are above this, the frequency changes is more than what we expect only by chance. But then previous experimental evolution studies have shown that long-range hitchhiking can play a very important role in inflating the number of SNPs that you identify as being under selection. This cartoon shows it really well, that if you imagine all these 12 positions or 12 regions of the genome, or 12 haplotype blocks, and then and then each row is one haplotype. And then so the haplotypes that are under selection are identified by these arrows in front of them. And then they all have this purple block. And in all these haplotypes, the purple block is linked to this red block. So when these haplotypes increase in frequency, and at the end, for example, you have a situation like this, you see an allele frequency increase, not only for the region that is under selection, truly selected, but also in the linked region. And of course, these two regions are separated by many different genomic regions that are not 100% linked between the two regions. So to account for the linkage, we reconstructed the selected haplotype blocks from the single SNP data that we had in our pool-seq. So this is based on, of course, if you have start your experimental evolution or study based on haplotype, already known haplotypes, meaning the sequences are already available, you really don't need to worry about this. But if you start from outbreak population, you most of the time do not have the financial capability to sequence everything. So what we did is to reconstruct these blocks. So the idea is that if you have your selected site at this region that is shown by this bar here, so the frequency of selected SNP and the hitchhikers would have correlation across time and replicates. So if you look for correlated allele frequency changes in your data set, you would be able to find as part of haplotype block that is under selection. So meaning that if this is the truly selected haplotype block, we would be able to reconstruct this region of it that is shown here. So you would be able to identify selected haplotype blocks. And the way we did it is that here I show part of Manhattan plot, the like the first 5 MB of chromosome 2L. So again, here we have chromosome positions, and then in Y we have negative log 10 p-value of CMH test. So we looked for a correlation between trajectories of SNPs across different replicates that we have and across time. So now we used all the different time points that we had. And then here you see different small haplotype blocks that have correlated frequency changes in their SNPs are depicted with different colors. So looking at, for example, at allele frequency trajectory of SNPs in this haplotype block that is in orange, you will see that they're of course correlated. Each box shows trajectory of SNPs in one replicate. In the x-axis, we have generation from 0 to generation 60, and in y-axis, we have allele frequencies. So I think the two important things that you can get from here is that first, all of them are correlated. They increase or not increase in frequency together, and there's heterogeneity across replicates. You see clearly that replicate 4, 7, 10, and at some point, I guess a little bit replicate 3 show increase for this particular region, but in other replicates, although they're still present, they're not lost, but they do not increase in frequency. So we looked at frequency trajectory of adjacent haplotype blocks, meaning that all other color-coded SNP groups. And then here in the plot below, you see, so each line shows the median allele frequency trajectory of SNPs in each of those small haplotype blocks. So a good replicate to look at is replicate 4, 7, and 10, that you see that despite the heterogeneity in the starting frequency of these haplotype blocks, all of them still increase in frequency together. And in case of, for example, replicate 10, at some point around generation 30 or 40, they all plateau. So that gave us this hint that these are all linked together, and then they belong to a much larger haplotype block, which spans around 3 MB here. So, and when we look at the frequency of this haplotype block, we see that from generation 0, which is shown with black color, to generation 20, 40, and 60, which is shown in green, the whole haplotype block increases in frequency in this span of 3 MB. So using this, we were able to identify 99 haplotype blocks, and then they range in size between several KB to several MB. So we have around 25% of these haplotype blocks that are larger than 1 MB. So from now on, I would be referring to these selected haplotype blocks as selected alleles. So some of the characteristics of these alleles were starting frequency, that the median or mean allele frequency is 0.18, and majority of these alleles start from low frequency. And then selection coefficient is also mean is 0.05, but we still have some alleles that are under strong selection. So what we were quite interested in was the genomic heterogeneity that we observed among these replicates. So to show heterogeneity across replicates, we came up with a summary statistic that we call replicate frequency spectrum. And I think this is something that you can do when you have multiple replicates available. So this replicate, the replicate frequency spectrum, reports the frequency distribution of replicates in which selected alleles increase in frequency. For example, here you see that 4 alleles increase in 1 replicate only, 8 alleles increase in 2 or 3 replicates only, and so on and so forth. So in this plot, you see that majority of alleles increase in 5 or 6 replicates, and there's only 1 allele that has consistent selection response in all 10 replicates. And of course, here to determine whether an allele is selected in a replicate or not, we had this threshold of 10% increase in frequency change. But regardless of threshold that we have, whether we increase it a little bit or whether we adjust it based on the starting frequency and selection coefficients of particular allele, we still observe heterogeneity across replicates. So what we were then interested in was to see what kind of evolutionary model would explain the genomic heterogeneity that we've observed across our replicates. As you can imagine, if you perform an artificial selection experiment and you select for a particular trait, for example, body size, number of seeds, or whatever you can do, then the trait is really obvious. So then you know when the optimal trait has been reached. So then it is much easier to analyze data. But this case is a case of laboratory natural selection, meaning that we don't determine the trait optimum. It's just the natural selection does its magic in the lab. So to be able to discern between the different evolutionary models, we use our summary statistic, the replicate frequency spectrum, meaning we use the genomic heterogeneity as a criteria to see how different evolutionary models fit the empirical data that we have. So initially we started with a selected sweep model, and for these types of simulations that I show, so we perform round of simulation for 10 replicates similar to the empirical data that we have. And for starting frequency, for selection coefficient, we all use information from the empirical data. And for each round of simulation, we then compare, we compute replicate frequency spectrum, meaning how many alleles increase in how many replicates. And then when you we repeat this 1,000 times, and then we can have, so these gray bars show 95% confidence interval of 1,000 round of simulations. So, and then these pink dots show the replicate frequency spectrum of the empirical data that we have. So here you see that the genomic heterogeneity really doesn't fill the sweep model because the difference is quite a lot. So, and then here we also, or I forgot to mention that we already included a linkage between the different alleles. So they are not freely recombining, they are linked, and the linkage mimics the linkage of the founder haplotypes. So I mentioned that we have this genomic heterogeneity, meaning that from the 99 haplotype blocks that we identified in these 10 replicates, not all of them show signature of selection in all replicates. And here in this plot, I show the number of alleles that we identify in each of the replicates, the 10 replicates. So on average, from 99 blocks, each replicate has 53 selected alleles. So that would mean that roughly half of the alleles that we identified responds in every replicate. So that means that we have quite low parallelism, low shared selected alleles across replicates. And you see that in this plot where it shows the pairwise similarity using Jacquard index. So if you have complete overlap, Jacquard index would be 1. And if you have 0 overlap, it's going to be 0. So the average pairwise Jacquard index is around 0.42, and then the maximum pairwise that we see is around 0.57. So we do not have a lot of similarity, a lot of parallelism across the 10 replicates. And then since we already have done phenotypic analysis, then we compared the phenotype of these 10 replicates for fecundity, for metabolic rate, and for lipid content. And we did not identify any significant difference between the 10 replicates. So meaning that all the evolved replicates have phenotypically converged. So now we have a case that all the evolved replicates have converged to the same trait optimum, but we have genomic heterogeneity. They have done this adaptation through different subset of alleles. And this is exactly what I introduced, that is one of the main characteristics of polygenic adaptation, that you have genetic redundancy. Then in one replicate, adaptation can occur via a subset of alleles, and then these subset can basically not respond in another replicate. So we tested this redundancy model by a very simple deleted jackknifing. So I have 100 selected alleles, and I know that these 10 replicates that I have, each of them would get around, on average, 50 or so selected alleles. So I randomly distribute them, and this distribution is basically blind to the identity of these selected alleles. So I repeat this, and then I have these 10 replicates, then I can compute our summary statistic, which is replicate frequency spectrum. And if I repeat it 1,000 times, I would be able to have this confidence intervals that are shown with this blue color. And then you see that this very simple full genetic redundancy model fits the genomic heterogeneity in our empirical data much better than sweep model that we checked before. And then when we observe that redundancy fits our data, then we tested for quantitative trait after a shift in trait optimum, classical polygenic adaptation. So that would be then, this is a fitness function that correlates fitness of population to the phenotype, and this would be the new trait optimum, and initial population would be, for example, somewhere here, and after 60 generations, it should move towards the new trait optimum. And when you perform this simulation, 1,000 round of simulation, so the green bars here show the 95% confidence interval of quantitative trait model with linkage, and this actually fits the genomic heterogeneity of empirical data much better than a sweep model. So using that, we compared different models. As I explained, we compared selected sweep with and without linkage. We checked a full redundancy model, and we compared quantitative trait model with and without linkage. And we saw that quantitative trait model and redundancy model fits the empirical data much better than the selected sweep model. And if we also look at parallelism, which is directly affected by genetic redundancy, across these models, we see that under a sweep model, we would expect higher parallelism, more similarity across replicates than under redundancy and quantitative trait model. And the parallelism that we observe in our empirical data fits better a polygenic adaptation under quantitative trait model than a selected sweep model. So for the summary of this section, so we observed that natural simulants populations, they have a vast reservoir of adaptive alleles that facilitates this rapid adaptation. And we did not observe parallelism across replicates. So we had this genomic heterogeneity, which actually fits polygenic adaptation well. And then the genetic redundancy that we observe, it allows multiple pathways that would all lead to convergence at a phenotypic level. And we did not find evidence of strong genetic constraint. So as I explained, polygenic adaptation and selected sweep that are normally contrasted against each other, but in fact there are extremes of a spectrum, and then there can be some cases in between that I actually presented, that we had a case of polygenic adaptation, but the allele frequency changes are not subtle, they are prominent, and then we could observe them in our experiment. So we used the summary statistic, replicate frequency spectrum, to differentiate between different evolutionary models to see whether our data set fits better under a sweep model or a polygenic adaptation model. But we were interested to find more of these distinct features between models that we can apply to small populations that we normally have in experimental evolution setups. And I have to explain that there are already different types of approaches that you can use to characterize polygenic adaptation in natural populations or in general. For example, you can look at correlation of SNP frequencies in multiple populations to environmental variables, or you can use gene ontology categories or pathways that, depending on that, each of the contributing SNPs would be weighted. And so SNPs and genes of a given category will be treated jointly, of course, and then the significance is tested by comparing them to a background set of markers. So, and then they would be, so you would identify significant SNPs or pathways. But in this method, you don't really have a direct link to phenotypes. But nowadays, especially with availability of huge GWAS data sets for humans, when you have phenotypes and genotypes available, there has been series of methods that can be applied. And then this is, for example, you can have a group of SNPs with significant association, and then normally with a focal trait. For example, it can be human height or waist to hip ratio. And then this difference should be basically, this difference should be statistically different from the background SNPs. And then this method had been implemented, and then signs of polygenic adaptation in some traits in humans have been identified. But there's this case of course, missing heritability, that much of the phenotypic variation cannot really be explained by the identified SNPs. So there are methods have been developed that they don't take into account only the significant SNPs, but all the available SNPs, and they have been, and then they are weighted according to their contribution to phenotypic variation. And then they have been successfully applied to traits such as body size or skin pigmentation in human populations, and features of polygenic adaptation have been identified. But what we were mostly interested in was to find, since we don't have huge GWAS data sets in our experimental setup, to find what are the features that we can look at that are different between these evolutionary models, and we can apply them to our experimental evolution setup. So for that, we performed a simulation study that we simulated SWIP and polygenic adaptations. But when we took advantage of different population sizes, we saw that there would be differences in different population sizes. And this is something that we can implement in the context of experimental evolution. So for the SWIP model, we performed simple forward right Fisher simulations. And for polygenic adaptation, we define polygenic adaptation as adaptation of a quantitative trait to a new trait optimum, where average the trait optimum should be shifted to this new optimum. So we define our trait, trait is affected by diallelic loci, and the effect size of the plus alleles are positive A, and then the frequency is P, and then the negative, and the deleterious one would be negative alpha effect size, and then the frequency would be 1 minus P or Q. And then in our simulation, we also assumed codominance. And so the trait Z was actually determined additively. And then we mapped our trait to fitness using a Gaussian fitness function. And so we simulated with a really simple scenario of 100 contributing loci, with uniform starting frequency of 0.05 and uniform effect sizes. And we had, as I mentioned, two populations, one small size of 450 diploid individuals and one 9,000. And then all the alleles are linked, and they are recombining according to the recombination map that we have from simulants. So we identified specific features that actually differentiate SWIP and polygenic adaptation in ENR experiments. So one of them is adaptation of fitness or phenotype, which is I think really straightforward and easy. So for having this information, you really don't need any genomic data. It's just only based on fitness or phenotypic data. So these in the x-axis we have generation, like 440 generations, something that you may be able to do in several years in during experimental evolution. In y-axis we had with a fitness or phenotype. So the green colors from now on correspond to sweep model, and then the purple one to polygenic adaptation. And then so the dotted lines show bigger population of 9,000 individuals, and the solid lines correspond to smaller populations. So we see that the fitness of population under sweep continues to increase, of course, until we have fixation of all the alleles or increase in the fitness. But then for polygenic adaptation, we have this increase in phenotype only until the new trait optimum is reached. And after that, phenotype does not change. And also the difference between the phenotype or fitness of small population and big population is more pronounced when we have sweep model compared to polygenic adaptation. Another pattern that we observed, and then they're different between these two models, is the dynamics of allele frequency changes. As I mentioned before, we have different stages of adaptation for polygenic adaptation, so that initially we have this directional phase until the trait optimum is reached, and after that we have this phase that can be short or long depending on the size of population, where drift disturbs the allele frequencies, and then after that some of the alleles would be fixed or some would be lost. So this is where we have sorting phase. So we can actually use the information from these three different phases to differentiate polygenic adaptation from sweep model. So here each row shows different generation. Here we have generation 10, 50, 100, and 140. And again, the left columns correspond to sweep, and the right columns correspond to polygenic adaptation. So what you basically have to look at is these stars, because they show the median allele frequency. So for sweep simulation, sweep model, you have consistent allele frequency change and subsequent fixation of alleles throughout the whole experiment. But for the polygenic adaptation, this frequency increase only occurs until the new trait optimum is reached. And after that, the median allele frequency actually plateaus. It really doesn't change. It is a little bit different in smaller populations because drift is quite strong. So you would lose some of these alleles, and then so they would be canceled out. So you would see a slight increase in frequency, the median frequency. And for this, of course, you need time series pool-seq data to be able to estimate frequency of SNPs in your population. And another pattern that we observed, and is different from between the two models, is parallelism across replicates. And that is, of course, you can imagine if there are sufficient genetic variation in the founder population, in case of polygenic adaptation, not all of those alleles are needed. Only a subset of those would be enough so that the population reaches this new trait optimum. That produces this genetic redundancy, and that produces non-parallelism across replicates. So here in these two darker colored lines, these show the parallelism in large populations, whether in sweep or polygenic adaptation. So in large populations, because drift is not really that strong, there is high parallelism across the replicates. But it becomes quite interesting when the population is small. You see that in this light pink color, where this is polygenic adaptation in a small population, similarity increases up to the point that trait optimum is reached. But after that, when this drift plays an important role, and after that, when we have this sorting phase, then some of the alleles are lost. So because of that, parallelism, meaning the number of shared alleles across replicates, decreases. While in fact, under a sweep model, you expect an opposite pattern, because generation after generation, more of the alleles increase in frequency, and you have more chances to identify those. So this parallelism is directly affected by genetic redundancy, and which is affected by the availability of genetic variation in the founder population. And another pattern that we observed is a haplotype structure. And that is, of course, in the during the course of adaptation, because of recombination, beneficial alleles would recombine, and then they would cluster on haplotypes. Under a sweep model, because adaptation continues to occur until fixation of these alleles, so you have consistent recombination of beneficial alleles. So the number of alleles, beneficial alleles on the same haplotype, increases throughout the whole experiment, whether it would be a small population or here a big population. Of course, in a big population, you have chance of identifying more of these alleles. But when we have polygenic adaptation, we have this recombination, but as soon as trait optimum is reached, this number would not really change. The number of beneficial alleles on the same haplotype would not increase anymore. So we have this plateau, unlike the pattern that we observe in sweep model. So of course, as I explained, that sweep, that polygenic adaptation are extremes of a spectrum. So of course, when you come kind of like in the middle of this spectrum, there are some gray zones that you really cannot differentiate these models. And one of them comes when the effect size of contributing alleles is small. In this case, even if you have a sweep model, because it would take a very, very long time until all the alleles are fixed, more than the duration that you can run your experiment in a lab. So you would not really observe the pattern of fixation that you expect under sweep model. So in this case, it has the allele frequency dynamics resembles polygenic adaptation. And also the opposite is also true. For example, if you have a polygenic adaptation, but in the founder population, you don't have enough genetic variation, meaning that you do not have redundancy. With lack of redundancy, now the population cannot really afford to keep some of the alleles of adaptation market. So all of the alleles need to respond to selection, to adaptation, so that the population reaches this new trait optimum. Or another scenario might be if the new trait optimum is really far, meaning the selection is really strong, then all of these alleles would get fixed until the population reaches this new trait optimum. So in this case, although you have a polygenic adaptation, but this signature of allele frequency changes that you see really resembles a sweep model. So in this case, we really cannot distinguish between these two cases. But if contributing alleles do not have, are not all small effect sizes, if founder population is not really limited in terms of adaptive genetic variation, and if the new trait optimum is not really, really far, we would be able to differentiate these two models. So as a summary, so there are temporal changes in allele frequencies, phenotype, haplotype structure and that parallelism that are that provide this information that we will be able to distinguish different evolutionary models, in this case specifically sweep and polygenic adaptation. And then machine learning approaches can be used to actually exploit these features to have a more quantitative approach rather than just a more descriptive one that we just did. And we think that actually by combining the analysis of large populations and small populations, we would be able to kind of like get the best of both worlds and then combine these signatures to be able to discern the evolutionary models much, much better. And for this, as I mentioned, I'm starting my own group 3 months ago, and we've started experiments to test the simulations that I just explained. So now, instead of 9,000 diploid individuals, I decided to just go big or go home. So we have populations of 100,000 diploid individual flies that normally we keep flies in populations of 1,000, and then normally they're maintained in bottles of 250 ml. So now here we have cages that are 60 by 60 by 120 centimeters. So they're pretty large, and then we can maintain 100,000 individuals in those. And then so we compare these large populations to small ones, which we only have 780 individuals in those. So, and then we maintain them under laboratory natural selection with the help of 2 technicians. And then, so that would serve as small and big population. And then we are planning to have time series genomic, phenotypic, and haplotype data. So for genomic and haplotype data is pretty easy. And for phenotypic data, we are planning to have series of different phenotypes from metabolic rate, body fat, or sugar, RNA-seq data, and also some life history traits from fecundity to longevity, and also body size, activity, and like resistant to different environmental stressors, including starvation. And I hope that by combining all this time series phenotypic data and genomic data in small and big populations, we will be able to identify, to basically corroborate the features that we identified in our simulations, or maybe identify new features that we can use for differentiating different evolutionary models and come up with methods to identify them in these populations. So with that, I would like to thank all the people who contributed to this throughout the past few years. So Christian Schlotter, who has been my postdoc supervisor and supervising most of the stuff that I presented, Joachim Hermisson, and a lot of people from the Institute of Population Genetics, and the 2 technicians that are helping me in the new project now, and funding agencies. And with that, I would be happy to take any questions.
Thank you very much, Neda. Thanks for this excellent presentation. Indeed, we will very soon move on to questions. But before we do, I wanted to remind everyone that we are recording this session and that it's also being streamed on YouTube. And so for anyone who would like to watch it later, it will be available on the EVOLTREE YouTube channel. And we are also welcoming any comments and comments on the presentation and the whole series in general, tagging EVOLTREE NW on Twitter, and also now during this webinar, you can feel free to tweet about it. So now regarding the discussion, you have an option which I'm sure you already are very familiar with Zoom, so there is no need for too much explanations. Anyway, we just ask you to raise your hand, and you will find the raise hand button under the list of participants, which you can access through the button at the bottom bar of your Zoom application. So I invite you, the floor is open to any questions. And we've got Moritz Ehrlich. I would also ask you to please, if possible, of course, to show your face. Yes, exactly. And also introduce yourself very briefly.
Hi there. My name is Moritz Ehrlich. I'm a PhD student at the University of Miami with Douglas Crawford and Margie Alexiak. And I'm one of the people who's trying to somehow find the polygenic signature in those natural populations. And yes, it's incredibly difficult. And I really like seeing, experiments presented like yours, where you can get much more of a grasp on the underlying mechanisms, because you have the lab-controlled environment. So I was wondering about something that you mentioned towards the very end of your talk, which is the fact that even though you might be in a selective sweep regime, you can still get this polygenic signal if effect sizes are small, and vice versa. You can get this sort of sweep signal in a polygenic regime when effect sizes are large or you're far from the trait optimum. So I was wondering what your expectations are, and maybe you've done this, when you have a distribution of effect sizes. So under a polygenic regime, I know there's a couple of studies that have looked at what happens in a sort of sweep model if you have a distribution of effect sizes, but I'm not sure anyone's done that for the polygenic sort of model, and what you would expect if there's both large and small effect alleles in a polygenic mode of adaptation.
That's a really good question, and I'm not sure if I have an accurate answer for that. But I think that, of course, what I explained in our simulation is just we had uniform starting frequency and uniform effect sizes, because we just wanted to have like a very simple simulation scenario to be able to find some distinct features. And even with these parameters, there are some gray zones that still might be problematic to identify this by just varying the effect sizes, even though it's uniform. So I know based on, I think the easiest one would be to just go to theoretical work, because for the theoretical work, they normally assume like an exponential distributions, that majority of them small effect, few large effect ones. But I can imagine that not sure about natural populations, but at least in our case, and the small population sizes, we would not really have the detection power to identify really small effects, effect loci. So the effect size should be more than what we normally discard as drift or by chance, that we would be able to identify that. And then even with that one, when I showed the S that we identified, we still had this exponential distribution. So I think this might be the case, and I think this is also what a lot of GWAS studies and human data sets have shown. But how this affects the patterns that you observe between these two, I think this is something that we definitely worth looking into this.
All right, thank you.
Thank you very much, Neda, and thanks, Moritz, for the question. I have seen very briefly Sally raising her hand. I'm not sure if, oh yes, but then you lowered it and now it's back up. So Sally, please.
Good morning or good afternoon to everyone in Europe. I'm Sally Aiken. I'm a professor at the University of British Columbia, so it's quite early in the morning here. I really enjoyed your seminar, very, very interesting. We work on long-lived trees, as do many people on this call. So we can't do the sort of experiments that you're talking about and can't even get close. We're lucky if we can look at a generation or two of selection, of artificial selection in breeding programs, for example. So mostly what we look at is using a space for time substitution when we look at the effects of environmental change on populations. So instead of changing the environment trees are in, we're looking at elevational gradients, latitudinal, climatic. And we do see strong signals that of polygenic adaptation rather than the sweep model. But I'm just wondering, there are all kinds of caveats that come with using space for time substitutions, and in the types of analysis that uh, you're doing, and when we look at genomic data in that context of space for time, and given that we have fairly high levels of gene flow as well, what kinds of cautions might you add, or opportunities? And I guess the second thing is I never thought of using a Jacquard index to look at those sorts of changes in allele frequencies across populations. So that's a simple approach, but that's quite effective. Anyway, I'm mainly interested in that space for time versus experimental.
I think I'll just go with this similarity index that we use. This is a quite a very simple one, and then I think the reason that we chose that is that because we really don't know, because it's possible that there are still a lot of adaptive alleles that they also contributed to adaptation, but we just did not identify them. So since we already knew all the adaptive alleles that we identify, so we could basically use it for comparison. But I think I can recommend that Sam Yeaman has come up with a much better similarity test that I think they call it C index, and I think it's much better for like gene expression data, for example, when you already know what are all the genes. So then you can take into account those genes that are expressed but not differentially expressed between the two samples or two replicates. So I think that might also be a really good alternative if you have a much bigger data set than just the 99 alleles that we had. And yeah, and for the question about the type of analysis that you can do using the genomic data in trees, to be honest, I'm not really sure if I have an alternative solution for that, because because most of the data sets that we have, we are not really constrained in terms of time series, but what it comes on top of my head is that I know there are groups that are actually using rings of trees as a form of time series data, and Kelly Swartz in GMI in Vienna has a really cool project on that. So I think maybe checking her website to see how she's doing it might be a good idea. I hope I've answered it.
So the tree rings let us look at responsive individuals within a generation. But again, we don't have that cross generation. So I guess another way to put that question is, if you had just come in at the end of your experiment and looked at your populations and looked at the genomic data for your populations in the warmer and in the original temperature, how much of this could you have learned from that single point in time, as opposed to tracking them over time?
Oh, you mean if we didn't have the time series data? Oh, okay. Actually, and I think this is the situation for many of the experimental evolution studies, because they, because of money or whatever, they only look at the founder population and the evolved one after several generations, or sometimes the populations, the experiment have started so long ago that those are not available. So they only compare different treatments. So I think for this one, definitely, this is something that you can do. For example, the CMH test that we did, we only look at allele frequency changes between 2 time points. And then for that test, we did not have the intermediate time points. But of course, this is, so for that, you would still be able to identify you would still, I think, be able to identify reconstruct alleles, but not haplotype blocks, but not really with much accuracy. But then I think if experimental haplotypes are present, that you can validate those haplotypes, it might help. But I think in terms of comparing across replicates and finding overlap, for that, you can even achieve this even without intermediate time points, because then you only need to, look at the last time point that you have, and then allele frequency changes throughout whatever generations. But of course, I have to admit, intermediate time points make the analysis much easier.
And in our situation, we don't even have the first time point. We've got a natural experiment with the last time point. But,
Yes.
But I think it's really helpful to understand what's going on with model organisms like your Drosophila, and to be able to then, reflect on our data for very different organisms. So thank you very much.
Thank you.
Thank you for that. Thanks, Sally, and thanks, Neda. We've got a comment or a question from Antoine. And before I hand over to Antoine, I just wanted to say that probably there is some technical glitch with the Zoom today, because we can't see the chat for some reason. And I'm not sure how to explain this fact, but all I can say is that I encourage you to raise your hands, and whatever you wanted to write in the chat, just to express it, and raise your hand. Be bold. Okay, Antoine.
Yeah, thanks, Neda, for a great talk. I have a few questions regarding the experiment on simulants. So you were looking at redundancy, of course, between the different replicates, but there might be as well redundancy, temporal redundancy between generations. Is this the case, really? Within a replicate, if you compare allelic changes from, let's say, generation 10 to 20, then generation 30 to 40, are similar alleles changing or different ones, or what's the amount of temporal redundancy?
That's a very good question. And after I finished analysis of this data set, actually a PhD student looked at this, and then I think that I can just send you the paper. So what they observed, so what they did is that, so since we already know what are the haplotype blocks, and then we know the frequency changes across replicates. So what she did is that she looked at to see how many of them she would be able to identify in the first 10 generations, how many in the first 20 generations, and so on and so forth. And then she found that there are few of them, maybe like 10% of these blocks, that they were only like she could only identify them at generation like after the first 10 generations. Mm-hmm. And then, but the rest of them, they were not identifiable in the first 10 generations, meaning that the allele frequency change was subtle until then, and it was only availability of later time points that allowed frequency change to be above our detection limit that allowed such a thing. So it is possible that we have, like, if you can imagine, early birds, that alleles that would respond earlier, and that might be if their effect size is bigger, so that they would basically contribute to the deterministic phase of adaptation. And after the population is closer to trait optimum, then other alleles with smaller effect size would contribute, so that the trait optimum doesn't overshoot, or just because some of them are at low frequency and they still need to escape the drift phase to basically undergo this directional selection. So we do observe such pattern.
One way to look at that may be to look at the temporal autocorrelations. Mm-hmm. When we have a time lag, generation lag, look at the correlation with allelic shifts between generation 10 and 20, 20 and 30, and see how this correlation evolves as you increase the lag time.
Okay, I see.
That might be a way to look at the temporal redundancy.
Thank you for that.
And about the second part, I mean, when you compare to the different models, what about the maintenance of genetic variation? Did you look at the genetic variation of the threat? It looks like the sweep model is not able to keep variation in comparison to the quantitative model. Is that the case?
May I ask what you mean exactly by maintenance of genetic variation?
The genetic, the additive variance. How does the additive variance evolve in the two cases in the two models?
I see.
At least when you look at the allelic profiles that you showed, it looks like there are more alleles are more maintained at intermediate frequencies in the quantitative model. Is that the case?
To be honest, I have only looked at to see whether the alleles are above our detection limit. And so we do have this, okay, I think I do have, oh, you mean what is the basically site frequency distribution between the sweep?
Look at that, well, then we may say something about the variance.
Yeah, I think if you want, I can share my screen again. I think this is what you to share. Okay. Yeah, I think this is no. Oh,
one.
Here, yes.
Yeah, this one. Yeah.
So, so your question is that about the variation in the genetic variation between the two.
Well, it looks like in the red case, I mean, you have alleles are more maintained at intermediate frequencies than in the green case.
Of course, I think because in the under the polygenic model, the trait optimum is actually reached at generation 40. So after this, what we have is basically drift and basically fixation or loss of these alleles. So from there, and the genetic variation actually decreases. So that is why you have a majority of them are at like low frequency. But in case of sweep, of course, is the opposite because they increase in frequency, and then as they the frequency increases, you have more power to detect them. So that's why you have this shift towards high frequency ones. So I think it's actually a really good idea. You can actually use the distribution of
calculated
site frequency to look at this. Yeah, thank you.
Thank you very much, Antoine.
Yeah.
I see that Christian is raising his real hand.
Yes. Thank you for the nice talk. Out of curiosity, I have a question to a remark you made. You said that you remove linked SNPs based on the founder population in the first part, as far as I remember. I was just wondering, have you ever looked at linkage patterns across time? And do they change? Because we would expect that if you have adaptation, that linkage changes at some places in the genome.
Yeah, so, okay, it has been my mistake if I said remove it, account for it, basically.
Account for it, yeah.
Yeah, for not focusing on SNPs, but focusing on haplotype blocks. And you're right, as we look into more advanced time points, linkage disequilibrium, basically the long range LD increases. And that is, we think, because adaptive alleles, basically adaptive haplotype blocks, they recombine on the same haplotype to be featured. And then because of that, it basically LD increases throughout the whole experiment. You're right.
Thank you, Neda. So are there any other questions? There is still a few minutes left. So in case you have any other questions or comments, please feel free to raise your hand. Oh, Christian again.
Yeah, I take the opportunity, a comment that Sally made just before. She said we don't have these nice time series experiments in trees. I don't completely agree. We could, and we do that actually, and some other groups do that as well. We can work with age cohorts because we have the tree rings, we can determine the age of each tree. And we can actually work with age cohorts and determine allele frequencies within these cohorts. It assumes that the selection pressure has happened rather early in the life stage of a tree, but we know that this is true. So we don't have these controlled time series experiments. That's what we don't have. But I think we have the possibility through the endochronological methods to follow allele frequency changes over time. And yeah.
Okay, Antoine, please.
I may comment on that. I'm working on oaks, and oaks are very long-lived. And actually, I have currently an experiment where I have cohorts that are 360 years old, other ones 205 different cohorts. And this goes back that these trees were born, and there was what was called the Little Ice Age, when it was much cooler in Europe than nowadays. And we checked at allelic frequency trajectories across ages, the different cohorts. And in 3 different replicates, 3 different forests. But it's all redundancy. No, you never find the same. Of course, there are many alleles changing. So we were looking, for example, at alleles that systematically increase frequency since 350 years to today, okay? And those they systematically decrease, and those they changed according actually to different temperature regimes. But in the tree, this was done in 3 different forests where we have these cohorts. And it's all different alleles and actually also different genes. We were looking if it were different pathways in different, there's nothing consistent across replicates. So just.
But is the environment also the same in this?
No, the environment is not the same. It's much cooler. We have records, actually, temperature records going back to about 500 years. So we have records of the time when these trees actually were born, so to say. And they are mostly selected because the highest selection goes on in trees at the young stage. So trees that are today, for example, you have a forest that is 400 years old. They were mostly selected about 380 years ago, because 90% of the trees, of the seedlings, are eliminated at a very early stage.
Yeah, so we did a similar thing with 7 replicates, and we found consistent allele frequency changes. But we only had 2 groups, so old and young trees. So probably then it's a bit easier. Of course, there are not many places in the genome that you find that change consistently, but we found.
And then, of course, you may have generation overlap. That's in the same, because these are in the same forest, these cohorts, of course. So they're not genetically independent. That causes an additional theoretical issue.
But I think, Anton, your observation is really, really cool. It basically goes to the genetic redundancy.
Oh, yeah, absolutely.
It just, I think the problem now is to find what is the shared selected trait or phenotype between them. But it is really cool.
But we looked, for example, at the annotation of the genes, where you had and thought that maybe these genes are involved in the same pathways, and it was not cool. It didn't work.
But it's possible that a higher level trait is actually selected, and then what you observe is just different gene expression or allele frequency changes at the lower level. So at the end, they might contribute. They basically the same higher level phenotype or intermediate level phenotype is selected, but through different pathways, which is really difficult, I think, to identify.
So it's a really challenging issue. But as I'm going back to what Christian said, there is possibility of doing what's diachronic approaches in and then not and then if you go to ancient DNA, then it will be even more interesting. Okay. One last question: in your experiment on simulants, did you follow the pedigree across generation? No.
No.
There might be a possibility to make some kind of genomic prediction. You calculate, estimate the genetic value, breeding value, using this in your population, and then look how the breeding value of the threats, the genetic value of the threats in the purely quantitative genetic approach, how the genetic value changes over time. But then you have no molecular signal. You take the whole genome as a predictor. You don't try to find footprint. You take the whole information. So.
Yeah, if you think about polygenic adaptation, and then there are many, many loci, then I think taking the whole genome would do the work.
Okay.
Okay, are there any other questions or comments? We still have time for one.
Sally, I see, has a question.
I see that Sally has shown herself, but yeah, sure, Sally.
As long as no one else does who hasn't spoken already.
Please go ahead, Sally.
So I just wanted to say that Antoine's comment about genomic redundancy, we absolutely see that. A huge amount of redundancy. Ian McLaughlin's on the call, and he looked at changes both over population. He looked at allele frequency differences across populations in different climates, and also with breeding, with 2 generations of breeding. And there are many, many loci involved, and only a subset of those has to respond in any population to generate the phenotypes that are associated with different climates. So I think it's really fascinating. And of course, that redundancy is a much more flexible system over time. So I think it's really exciting that we're seeing all those differences. And Christian's absolutely right. Cohort studies are useful, but we can't do them over tens of generations. And that's the piece that we're really missing. So, but beautiful talk. Thank you.
It's another kind of plasticity. Yeah.
Okay, so Antoine, would you like to give some concluding remarks?
Well,
because this is the last of the episodes.
Okay, thank you, Neda, for your presentation today. It was really great, and I would like to take also the opportunity to thank all the people that attended these sessions. And I guess we will renew it in the future. We will have discussion on that, but I think it's really a nice occasion to meet while we are all on lockdowns in different countries, and we need somehow to communicate, and this was a very good way to do it, and on top of it, on very good science. So thank you, all of you, both the speakers and all the attendants.
Thank you for Antoine. Nice. And I just wanted to remind everyone that this session was also organized in collaboration with Stephen Cavers from UKCEH, Christian Rellstab, who is here with us today, WSL, and Santiago Gonzalez Martinez from INRAE. So thank you also to them for putting this whole series together, and to Antoine for kindly agreeing to moderate and to be here with us. So we really appreciate that, Antoine. And thank you, Neda, for being with us today. I would like to also thank the online audience. And as I anticipated, we will be sending out a short survey to evaluate whether you enjoyed this initiative, and we might perhaps renew it next year. But for now, I would like to thank you all and wish you a nice evening or morning or day, wherever you are.
Bye-bye. Bye, everybody.
Bye. Bye-bye.
E.